our attention to a panel discussion exploring the evolving role of stem cell therapies and how they are revolutionizing targeted healing and regeneration. And for this discussion, please do welcome to the stage Dr. Shahrukh Hashmi, Director of Research at the Department of Health, Abu Dhabi, Dr. Yendry Ventura, CEO of Abu Dhabi Stem Cell Center, Neil Warma, President and CEO of Mongoose Bio USA, and of course your moderator, Dr. Wisam Ismail, Consultant Oncoplastic Breast Surgeon at American Hospital, Dubai. Please put your hands together as we welcome them to the stage.
Right, good afternoon. So I'm very privileged to be among a great world-renowned scientist, actually. So yeah, I'll talk the least because we need to listen to the great guys. Okay, so yeah, Mr. Neil, all yours. So I think I have a question in mind. What do you think is the future of MSc? And that's for the whole panel.
Leading question. Well, thanks to the organizers first for inviting me and putting me on this stage with such esteemed guests. Just a little bit of, I guess, a background as to kind of where I see some of the breakthroughs in cell therapy. And just to give you 30 seconds on kind of my company. So we're a cell therapy company. We're focused on, really on the immune system, kind of retraining the immune system to fight cancer and to fight aging. And for me as the CEO and kind of the founder of the company, what's really important to me is that we're developing therapies for cancer, for aging, that are truly differentiated. I think it's important for any company to have a differentiated asset, a differentiated technology. So in the cell therapy world, and this company was spun out of MD Anderson, the largest cancer therapy company really in the world. And this was based on kind of my scientific founder, Dr. Cassini, 20 years of research. And he came at it from a perspective of what's going to really break through with cell therapy. Where are the challenges that we're seeing in cell therapy? And this is really the premise that we spun Mongoose out of MD Anderson. And there were really two challenges that we saw in the cell therapy world, in the cancer treatment world. And the first one being kind of the lack of novel targets. And we know that CAR T's been around for a number of years. We know it's been successful, but there's limitations with CAR T in being able to identify and access novel targets. And we kind of look at it as kind of an apple. And the targets for CAR T are the peel or the skin. And the number of patients, cancer patients that can benefit from CAR T therapy are less than 3%. So the challenge was, how do we identify novel immunogenic tumor targets, kind of the pulp of the apple, that intracellular target. So that was one of the first challenges, and we have a very powerful discovery platform where we've discovered thousands of potential targets, and we've licensed 4 of the most relevant and validated targets into Mongoose. The second challenge was understanding and being able to, when the immune system kind of attacks cancer, when you give a therapy, the T cells, the immune response is very potent, but it's relatively short-lived. We know there's a potent response, but after 4, 5, 6 months, the T cells become exhausted, and the cancer comes back and the patient dies. So the other challenge that we looked at within Mongoose was how to prevent T cell exhaustion. How to make T cells, or memory cells specifically, persist over time. And that technology we've worked on over years to be able to reprogram memory cells, T cells, into persistent, long-lasting cells that will suppress the tumor. And we've done this in the clinic for years. So we hate to use the word cure, because that's a very strong word. But I think the two breakthroughs that we're bringing to the field of cancer, and similarly to aging, will be in identification of some very powerful discovery platform to identify novel targets, and this ability to retrain or reprogram T cells to persist over years. And all this is kind of wrapped together with a really potent AI system. Maybe I'll touch on this later, but AI has been used to screen millions of spectra to identify our lead targets. And we see our AI platform as providing us with kind of a night vision that allows us to identify these targets that remain hidden from the immune system. Cancer cells, aging cells are very good at camouflaging and remaining hidden. So our AI technology, as we like to think of it, gives us that night vision to go in and uncover these cells such that we can arm our T cells and attack the tumors and attack the aging cells as well. So that's a little bit how we see breakthroughs and next generation cell therapy coming out of Mongoose.
Excellent. Thank you very much for that. So yeah, Dr. Yendry, any comment on this? And you tell us a bit more about your company as well, please.
Yeah, thank you. Thank you for having me. I think Neil is right in the challenges. We have found similar challenges in our research too, especially when it comes to analyze, complex systems and complex behaviors like cancer and autoimmunity. Identifying proper targets is not really easy. It's complicated, and predict how efficacious that therapy is going to be is also really challenging. I think one of the success from Abu Dhabi Stem Cell Center point of view is implementation of a really translational view of cellular therapy, and the best example is the Abu Dhabi bone marrow transplant program, in which we not only implemented bone marrow transplant but also CAR T therapy. And recently we announced the expansion of our cellular therapy program with the isolation of tumor-infiltrated lymphocytes, and also working in TCR-T like Neil company is working, but in this case for autoimmune diseases. Now, what I think is really relevant is not only the fact that we have the capability to perform this type of alternative, but also the importance of other players and stakeholders coming together and helping to make these therapies not only a reality, but also accessible to the patients.
Yeah, I think behind the scene we were discussing, the need for the tumor cells and, how easy to obtain in this part of the region, in your opinion?
So I can tell you it's not easy because we are pioneers, and whenever you are a pioneer in something, you need to break a lot of barriers, especially when it comes to establishing new protocols and establishing new methodologies, right? However, I believe that we have the right infrastructure, the right ecosystem to do it, especially with the support of the Department of Health. They have been instrumental in accelerating, the registration of these, let's say, kind of methodologies or protocols that also is going to guarantee in the future that the therapy can be accessible, because R&D is not about discovering. R&D is about creating solutions for the patients that need it. And for that, you need to create a kind of awareness from the beginning that everybody understands, not only in the R&D center, but the hospitals, the payer sectors, and the regulators.
Yeah, definitely. That's really great. So coming to Dr. Hashmi, again, we're discussing, what's your views, on the, the autologous and allogenic Arctic. So, that's a big topic, and I'm sure you're authority about that. So, yeah, the stage is yours.
Well, thank you. The question is different. The future allogenic versus autologous CAR T cells, or even CAR NK cells. The current 7 FDA-approved CAR T cells are all autologous, 2 for BCMA and then CD19, leukemia, ALL, and then for lymphomas. The allogeneic CAR T field is not new, actually. They've been producing these preclinical models for more than a decade now, and now the phase 3 trials have started, and these are alpha, beta knocked out by gene editing, right? And the gene editing techniques have improved from CRISPR-Cas9 to base editing, to prime editing, base editing for SNP, for single nucleotide base disorders, and the prime editing for complex genetic mutations. So we've done a good job, many groups in China and US and many other countries of the world. The day you get the phase 3 trial ended and the BLA submitted and accepted, it's very likely that the allogeneic CARs, if the safety is again proven and efficacy is proven, the whole field of autologous CARs may be affected. May or may not be, we don't know, we can never predict. But as Yendry mentions, how can we make it practical to get to the patients? And as Neil mentioned, it's all about not bench to bedside. This whole concept is wrong. I have to be provocative. Even word bench to bedside, because you're implying that chemistry, preclinical development, ADME, lead optimization candidate, preclinical, phase 1, 2, 3. Actually, it should be the other way around. From bedside, clinician says, what's the impact? What is the need? Sickle cell thalassemia, for example. Take it to the bench, the lab, and bring it back. And in order to do that, Department of Health is doing everything we can to have free INDs at the moment, IND discussions to even design the clinical trial and look at the preclinical data and organoid data, if that's available, very carefully, and to expedite this thing so we can get the phase 1 trial and then phase 2 and 3 trials together, hopefully shorter period of time to get at the patients. Because if it doesn't get approved, it doesn't help anybody. It doesn't help the patients, right?
Definitely.
I mean, you can publish a million papers, but it doesn't help the patients. So that's what we need to do.
Yeah, and in terms of time scale, roughly, for the audience, how long would that process would take, in your opinion, when it comes to designing, get like a phase 1 trial, that kind of things, on that sector?
Yeah, so it's different for monoclonal antibodies versus small molecules versus cell and gene. Now, this session is on stem cells, so cell and gene therapy, right? So the pre-IND meetings can happen fairly quickly, and if the preclinical models for IND-enabling studies are good enough, you should very quickly move to a phase 1 trial if you have got the target well. It's not mandatory for each and every single product to have 2 animal species. In fact, patient-derived organoids for cancers, as you well know, should be utilized as much as they can. Machine learning approaches for in silico drug development have to be utilized. Not many companies are doing it. We've known about AI since 1955, right? But how many companies have actually done that to take it to the human trials? Not many, actually. So we've got to use that. We've got to use our PDOs, patient-derived organoids, for testing. And then preclinical models, dose finding, and immediately. So instead of like 8 years to 10 years, I think we're hoping that in Abu Dhabi, that with clinical trials, that we start with the IND and IRB from the bench for an identified problem from the bedside, which should take 4 to 5 years rather than 8 to 10 years. That's what the hope is.
Excellent, that's great. Okay, so coming to Dr. Warma, we're discussing as well behind the scene, like how feasible it is to establish like a research unit, what you need for that. So what's your views on that, and how is the AI, you think, in your opinion, is going to help to speed it up?
Well, yeah, I mean, I know a lot of focus is on AI, and I think that can be a terrific facilitator. I think we have to be careful kind of promising the world with AI, because AI is only as good as the people that are interpreting the data, and it's only as good as the data that comes out of it. But there's huge advances that can be made in time and cost using AI as we do. I think it's really critical, as Dr. Hashmi said, that this time to patient, and it's an interesting panel. Certainly, the streamlined approach that's been taken here in UAE, Dr. Osman and Dr. Hashmi have just pioneered this, which is really, really impressive. And I think it's important to, for all of us, the different parts of the world, to learn from each other. Dr. Hashmi and I are talking about doing a panel at Bio next year to really bring together regions to educate each other as to best practice. And I think reducing this time from R&D to patient is absolutely critical. And interestingly, we have Yendry here with a CDMO on board, and a lot of the discussion we're having in the US is on the manufacturing side for cell therapy. How can we reduce the time it takes? Because all of us, autologous, we take cells from patients, we modify them ex vivo, so in our manufacturing facility, and then infuse them back into the patient. So what's very important, especially for very sick cancer patients, is how quickly can that process go? And now they're looking at in vivo CAR T and in vivo manufacturing within the body. So what we're doing at Mongoose, we've incorporated technology early on in the process. It's going to drive up yields. And what we're looking at doing is turning around, kind of having from vein to vein, as they say, so from the body back to the body within a week. So there's all sorts of ways of streamlining the process. AI can certainly help, but again, you need that knowledge base, a skill base, the expertise to understand how to leverage your AI and how to leverage the patients, and also how to learn from each other. I think that's why it's terrific for us coming from the US and spending time here and vice versa, just learning best practice. They're doing terrific things in Asia with the IIT trials, and we're looking at a partnership with the Koreans who have superb clinical trial expertise, and they can do things very fast with a regulatory process in South Korea. So there's a lot of learning that can be had amongst all of us, which is really key.
Excellent, thank you. So, Dr. Ventura, me as a breast surgeon, always my attachment to stem cells is when we do a liposuction and purify the fat and inject it where the cancer is as a like a one-stage breast cancer and reconstruction at the same time. There are some fears always that these new stem cells can, ignite, they can stimulate the breast cancer. How true or how wrong is that statement, in your opinion, as a stem cell?
That's a very good topic, and it deserves a full conference for that only. What I can say is, in my years of experience, I have never seen it. Okay. And I have many other experts in my laboratories with more years than me, and they also didn't see it. Now, especially when the cells are minimal manipulated or manipulated without genetic modification, for example. So the risk is really very minimal. Now, when you start using, for example, Yamanaka factors for the generation of induced pluripotent stem cells and therefore later differentiation into functional cells, now the risk is higher because some of those factors are related with cancer biology. So yeah, it could happen. And one of the ways that you actually prove that some of those stem cells are actually functional is by the generation of teratomas that tells you that they can actually be pluripotent cells or at some point even close to totipotent cells. But none of those cells are actually right now available for clinical use. That's the good part. So there has been a lot of work in R&D to prove the safety of those cells before they actually go for clinical trials. And I believe that even before that happens, we need to focus a lot in the functionality because there are a lot of challenges to generate terminal differentiated cells, let's say for pancreas or for kidney failure or for even from brain problems. So I'm very confident of the product we have right now and know that the risk is very minimal.
Yeah, definitely, because to my knowledge, there's no like a level 1 RCTs related to that. These stem cells can really affect the breast cancer when we, do the fat transfer, when it comes to the cancer bed, because the patient will have like all the adjuvant treatment between chemo, radiotherapy, biological agents, etc. And that really has not shown, evidence-based medicine that, yes, these stem cells can be dangerous and harmful on the cancer bed, that they can generate new cancer. So I totally agree with that. So, right, Dr. Hashmi, so what's your view about that as well?
Yeah, I would love to answer that question. Arnold Kaplan's discovery of the MSCs was 35 years ago, and at that time they were causing mesenchymal stem cells, not stromal cells. ISCT changed the name afterwards. We have tens of thousands of patients treated with autologous MSCs. And they're manipulated. There's a bioreactor right there in the OR. We do that for hips. Clinical trials. It should be on clinical trials.
Trial level, yeah.
Today, after 35 years, the only MSC approved in the world is actually for GVHD, acute pediatric GI GVHD. And same thing with TILs and TCRs. So when Steve Rosenberg, I'm sure Neil you'd remember, 1998 he started the first clinical trial, 1995 actually at NIH. And after 35 years, one agent approved for TIL, melanoma, one for TCR for synovial sarcoma. But we haven't seen, I have to agree with Yendry, these are manipulated, these are autologous. They don't have any other properties that would make them oncogenic. And in fact, those cases in which there was suspected cancer coming back, there was actually anaplastic lymphoma. And that was because of silicone implants. And actually, silicone implants for breast enlargement were no longer used too much because of the risk of lymphomas. And there you put anything, actually, your risk of lymphoma is going to be high. But breast carcinoma, adenocarcinoma, we haven't seen that at all. It's very safe. I don't think there's any question about it.
Yeah, definitely. And even what we call the BIA-ALCL, which is like breast implant-related atypical large cell lymphoma, this is mainly, related to like a specific kind of a shell when it comes to the breast implant, the rough shell, the bio shield that happens there and appears there, that it's going to cause a lot of like chronic inflammation, and then it might create that. So yeah, that's excellent. What's your view on that as well?
Well, I'll let the experts talk to that because I don't, as I said, they kind of answered it well, and I totally agree. Dr. Hashmi's got way more experience than me. The one thing, because I see we're kind of running short of time, the one thing I just want to kind of acknowledge is, as we go kind of forward with all of these, potential therapies and differentiated assets. I think it's really important to, and talk about, AI and such. I think it's really important to, let the data decide on our course of, our course of actions here. And I think we have an obligation, certainly to the patient. And one thing I always say, kind of as a CEO, I think our most important, our most important kind of activity or philosophy is managing expectations. And I think we have to be careful to manage the expectation of the patient, to let them know what's coming, to not raise or lower their expectations, but to be as factual as possible, driven by data. And then we also have an obligation to our shareholders. We've got investors that invest in the company, so they have to know what to expect, and we have to manage that well and let our decisions be driven by, real data. There's a lot of activities that are going out there. There's a lot of talk about AI. But again, it has to be data-driven to be able to manage those expectations, to be able to deliver drugs to patients, and deliver a return to the investors as well.
Definitely, yeah, that's a great statement. Dr. Ventura, 30 seconds of what's coming, what's the future in your opinion? And I'll ask the same question to Dr. Hashmi.
I mean, I can't wait to see the future. I cannot predict it, but for sure I think that there has to be a revolution in terms of regenerative medicine. I always say this, that we have been living since the inception of bone marrow transplant with BMT as the only really approved and trustworthy cellular therapy worldwide use, right? But there are many other things that has to happen. We have patients with chronic kidney disease, we have patients with pancreatic problems, we have patients with heart issues, and regenerative medicine need to go to that place, capable of not regenerating, but generating new organs. So we, in the future, can avoid this kind of a living donor transplant, and even disease transplant as well. So I think that should be the future, and I do believe that artificial intelligence is going to help us to get there faster. And when that moment happened, everybody, I think, is going to be very pleased and happy.
Excellent, thank you. Dr. Hashmi, same question. What do you think, where are we going? What's the future of stem cells, AI, cartels? So what's your views? What's going to happen in the next 5-10 years? I know this region is huge when it comes to research and development, investment heavily in AI, investment in stem cells and regeneration.
Yeah, this wasn't discussed before, the question of future, but I don't predict future at all. 90% of Nostradamus' predictions were wrong. All these predictions we hear of colonies on the moon, all this craziness, and the flying cars have not been proven. Majority of them are wrong even today about AI.
Totally agree.
But what I see the promising therapy for the people of the world is the in vivo therapies for sickle cell and thal and many other genetic diseases, because we need off-the-shelf gene therapies available, which don't require the complex management. I'm very hopeful that will happen, because they have not infiltrated Africa and Asia, where it's needed the most for sickle and thal. Similarly for the CAR T cells, allogeneic CARs, I'm hoping that's going to be promising in the future. Phase 3 trials are already going on for that part. For gene therapy, in vivo gene therapies, it started phase 1 already in many countries.
Yeah, correct.
I hope that's going to be true soon.
Excellent. I just like to thank the panel, and really best of luck, guys. Thank you very much. You're doing a great job.